This is the fourth video in the series of lectures on the cell cycle. In the first three videos, I generally told you about the different phases of the cell cycle. I've told you about the machinery that is required for the execution of the cell cycle and its component. And I briefly told you about the control system that is controlling the machinery of the cell cycle. We also discussed about the organelles, how these organelles they are going to get duplicated, and how they are actually segregated in the M phase of the cell cycle. Today we are going to specifically talk about the events that are happening in the G1 phase of the cell cycle. I've told you that the G1 phase is actually a decision-making phase of the cell cycle, and one of the important things that the G1 phase checks for is the availability of the growth factors. Now, if these growth factors, they are available, what they are going to do is they are going to bind to the growth factor receptors which are present on the surface of these cells. Now, the binding of these growth factors to a particular receptor is creating a cellular signaling pathway, and the cellular response to these pathways is that the gene expression is going to change in these particular cells. Now, when these gene expression change, of course, the concentration of different kind of proteins, they are going to be increased in the cell. And we will discuss the examples of the proteins that are going to increase in concentration in the G1 phase of the cell cycle. When you talk about the DNA, they have got these specific regions known as the origin of replication, which I have highlighted in orange. These origin of replication are the points from where the replication of the DNA starts. If you look at these origin of replications, to this origin of replication, a protein complex is attached, which is known as the origin recognition complex. Now, these origin recognition complex are attached to this origin of replication all the time. But the presence of this origin of recognition complex does not guarantee that this origin of replication will be creating the replication. They will be replicating the DNA in the cell. You have to add more proteins to this origin of replication so that you can actually fire these origin of replications. Now, when the growth factors, they bind to their receptor and the gene expression change, one of the important protein whose concentration is going to change in the G1 phase of the cell cycle is known as the Cd6 protein. Now, the concentration of the Cd6, when it increases, this Cd6 is going to bind to this origin recognition complex. So now you are adding extra proteins to these origin recognition complex. Now, once the Cd6 has bind to this origin recognition complex, it is actually creating a binding site for another protein which is known as the CDK-4 protein. Now, if you talk about the CDK-4, their concentration is not going to increase in the G1 phase, but they have been released from an inhibitor. And we will discuss that how this CDK-4 that is inhibited and how it is released or how it is activated in the G1 phase of the cell cycle so that it is able to attach to this origin recognition complex. So first you have attached the Cd6 to this origin recognition complex, and now you are adding the CDK-4 to this origin recognition complex. Once the Cd6 and the CDK-4 that has bind to this origin recognition complex, you are adding a ring complex to this, which is known as the MCM ring. Now this M, this M is actually mini, this C is chromosome, this M is maintenance ring. This MCM ring is actually made up of 6 proteins which are known as the MCM2, MCM3, MCM4 up to MCM7. This MCM ring, it is very important for the unwinding of the DNA at the replication forks in the eukaryotes and in the archaea. So when the Cd6 that has bind to the origin recognition complex, the CDK-4 that has bind to the origin recognition complex, the binding of these two proteins is actually creating a docking site for this MCM ring, which actually is going to bind to this origin recognition complex now. Now what you are doing in the G1 phase is that you are adding extra proteins to this origin recognition complex. And when you talk about the complexes of these protein, like when you talk about the binding of the Cd6, the CDK-4 and the MCM ring to the origin recognition complex, this whole complex is known as the pre-replicative complex. Now that means that in the G1 phase, you are actually preparing the cell for the S phase of the cell cycle. So the complex of the origin recognition complex, the Cd6, the CDK-4 and the MCM ring is called as the pre-replicative complex, and for short it is known as the pre-RC. So this is one of the events that is happening in the G1 phase of the cell cycle. Another important event that is happening in the G1 phase of the cell cycle, that the expression of another gene that is changing, which is known as the Cyclin D, and you are actually increasing the concentration of the Cyclin D. Now if you remember the control system, I've told you that these cyclins are the regulatory subunits for a set of proteins which are known as the cyclin dependent kinases. If you look at the cell cycle, in each and every phase you are going to find specific cyclins and you are going to find specific CDKs. So when you talk about the G1, the specific cyclin for the G1 is called as the cyclin D, and the specific CDK-4 for the G1 is called as the CDK-4 or the cyclin dependent kinase 4. This cyclin dependent kinase 4 is actually a serine threonine kinase. That means that it is kinase in nature. It can add phosphate group to its substrate protein, but this can only add phosphate groups to the serine amino acid or the threonine amino acid on its substrate protein. So this is actually a subset from the kinase family. Now when the cyclin D that binds to the CDK-4, the binding of the cyclin D is actually activating the CDK-4 in the cell. Now these CDKs or this particular CDK-4, its concentration is not going to change. They are already there in the cell, but they are in the inactive form because of the unavailability of its regulatory subunit. So once you have bound the growth factor to its receptor and the expression of the cyclin D changes, its concentration increases, they are going to bind to this CDK-4, thereby activating this particular kinase. Now one important thing is that the complex of the cyclin D and the CDK-4, that is called as the G1 CDK. Now the G1 CDK doesn't mean that you are talking about the CDK-4 only. You are actually talking about the complex of the CDK-4 to its regulatory subunit, which is the cyclin D. So once the cyclin D has bind to the CDK-4, which actually makes the G1 CDK. The binding of the cyclin D to its CDK-4 is actually going to activate this. Now, once you have activated this particular kinase, it is going to act on its substrate. Now, the substrate for this CDK-4 is another protein which is called as the Retinoblastoma Protein. Now, it is called as a Retinoblastoma Protein because it is one of the genes that is mutated in the retinoblastoma disease, and this Retinoblastoma Protein is actually a tumor suppressor protein. So once this is mutated, the function of it as a tumor suppressor protein is gone. That means that there will be tumor in the retina of the eye. Now, initially, this retinoblastoma protein is in the hypophosphorylated form. By that I mean that the retinoblastoma protein is in the phosphorylated form, but in the hypophosphorylated form. Some of its amino acids are phosphorylated, but not all of the required amino acids are phosphorylated. So we call it as the hypophosphorylated form, a less phosphorylated form. Now when the retinoblastoma protein is in the hypophosphorylated form, it is going to bind to an important transcription factor which is known as the E2F. And when the retinoblastoma protein, for short the retinoblastoma is known as the RB, so when this retinoblastoma protein is attached to the E2F transcription factor, your E2F is in the inactive state. This E2F transcription factor is very important if you want to move from the G1 into the S phase of the cell cycle. So if the E2F is in the inactive form, that means you cannot move from G1 into the S phase of the cell cycle. Your cell cycle is stuck in the G1. Now what you have to do is that if you want to move from the G1 into the S phase of the cell cycle, you have to activate this E2F by removing this retinoblastoma protein. Now what the CDK-4 is doing, that when it is activated, it is going to convert the retinoblastoma from the hypophosphorylated form into the hyperphosphorylated form. That means you are adding extra phosphate groups to this particular protein. Once you have converted the retinoblastoma from the hypophosphorylated form into the hyperphosphorylated form, there are structural changes going on in the structure of the retinoblastoma protein, and in this particular hyperphosphorylated form, it is not able to bind to the E2F. Now once the retinoblastoma cannot bind to the E2F is going to move from the inactive into the active form. You can actually move from the G1 into the S phase of the cell cycle. We will continue this discussion in the next video.